Hi, I'm Daria Zikaus, and today I will present to you the results from our paper entitled "D-Fos regulates macrophage infiltration against surrounding tissue resistance by a cortical actin-based mechanism in Drosophila." My lab studies how immune cells can infiltrate into tissues, a process that we call tissue invasion. Vertebrate macrophages do this. They migrate into tissues during development where they take up residence. In these tissues, they then can regulate homeostasis and can secrete factors to organize tissue repair upon injury. However, how they do this, what the mechanisms are that immune cells utilize to move through tissue barriers in living organisms, is not well understood. We wanted to know if a program that enabled tissue invasion might be induced in immune cells by the enriched expression of a particular transcription factor. We studied this in the simple model of macrophages in the Drosophila embryo, migrating between the two tissues of the ectoderm and mesoderm into the region called the germ band. First, we identified 12 transcription factors which are expressed in macrophages during germ band tissue invasion. We then focused on one of them, D-Fos. We chose it because it is the ortholog of the vertebrate proto-oncogene C-Fos, which is known to be involved in the invasion of cancer cells. Using live imaging, we found that D-Fos speeds macrophages' initial entry into the germ band tissue and their subsequent movement between the two tissues in the germ band in the first 20 micrometers. We used RNA sequencing then to reveal that D-Fos actually leads to the increased mRNA levels in macrophages of 10 other genes. These include the filament actin crosslinker, Cheerio, and the tetraspanin, which is a membrane-spanning molecular scaffold called TM4SF. Interestingly, expression of C-Fos in mouse osteosarcoma also leads to higher mRNA levels of the vertebrate filaments A through C and the tetraspanin ortholog TM6SF, arguing that Fos increasing levels of filaments and tetraspanins might be a conserved property in many organisms. To check if these D-Fos targets are themselves important for invasion in the fly, we used RNAi knockdown. And indeed, we observed that both the filament Cheerio and the tetraspanin TM4SF are required in macrophages for tissue invasion. But how might they be doing this? We found that both enhance the activity of the Rho1 GTPase, and thus the actin-assembling formin diaphanous, as well as increasing levels of cortical actin. We therefore concluded that D-Fos upregulates two cooperating molecular pathways to stimulate the assembly of actin at the cell membrane, which is also called the cortex. This effect on actin from D-Fos in the pathway is critical, since when we express a dominant active form of diaphanous that can induce actin polymerization, we are able to rescue the macrophage invasion defect if D-Fos is absent. But why is this effect on actin assembly important? Actin polymerization is known to be needed to advance the front of the cell and to bring the cell body back forward. However, a role for cortical actin all around the cell surface had not been previously examined. We observed that macrophages lacking D-Fos were longer and skinnier as they initially invaded, but not when outside the germ band. This suggested to us that the cortical actin assembled by D-Fos might help macrophages resist the pressure from the surrounding tissues. To test this, we asked what would happen if the surrounding tissues did not exert as much force on the macrophages during invasion. We decreased the stiffness of the ectoderm, and indeed, we were able to rescue the invasion of macrophages lacking D-Fos. While this was exciting, it was also surprising. In other systems, the stiffness of the nucleus, not the cortex, is what determines how well a cell invades through narrow spaces. Nuclear stiffness is affected by lamins, proteins whose expression make the nucleus harder to form and thus can, for some cells, impede cell invasion. We asked if nuclear lamins play a role in macrophage invasion in our system. Amazingly, overexpressing lamins or knocking them down in our embryonic macrophages had no effect on their capacity to invade. However, when D-Fos was no longer functional, then reducing lamins increased macrophage invasion and rescued the D-Fos defect. This argued to us that normally the cortical actin shell induced by D-Fos prevents the stiffness of the nucleus from playing a role in invasion. We hypothesized that the cortical actin induced by D-Fos could act like the outer walls of an ice-breaking ship, protecting the nucleus inside and helping push away the surroundings. This strategy could allow immune cells that sit in tissues and live for a long time to protect their nucleus and thus their genome from damage when moving through tricky environments. This should be explored for immune cells in vertebrates. Finally, this paper was due to the efforts of Vera Bilyeva, who initiated the project and obtained key insights and findings on the mechanism of FOS action with the help of Igor Gutschin and Attila Gyurgy. These findings were completed and extended by Steffi Wachner and Attila Gyurgy with the help of Shamsi Imtinani, Masha Akhmanova, and Marco Roblek. We are also very grateful to our collaborators at the Medical University in Vienna, Marcus Linder and Maria Zabilia, for their work on FOS in mice.